Now I'll show you how we download these polygenic scores or these polygenic risk scores from PGS catalog. How do we do that? So what we're observing here is that in PGS calculator, what we have is we have scoring files that have this following format, where it says PGS catalog scoring file, and it will tell you, where to find additional information, the format version, the polygenic score information, the PGS identifier that it has, the name, the trait reported, mapped, ontology trait ID, the EFO, the genome build, the assembly like GRCH38, which refers to HG38, GRCH37 refers to HG19. They do have another video going over these concepts. And then we also see the variance number, so number of variants listed in that file, that PRS file, PGS file, the weight type, and if it's beta, if it's odds ratio, if it's HR, the PGS publication identifier, citation, license. So these are some of the headers that we would expect for typical score files here, and they usually led with RSID reference SNP IDs. So that's fairly common. So we can go to the PGS catalog and then we can click on the polygenic scores. And then we'll see that there are a lot of different scores here that we can see. For instance, we can look at this one here, PGS 000018, which is for coronary artery disease, and it has like 1,745,179 variants here. That's a lot of variants for this heart disease, for the coronary artery disease here. There's a lot of variants that they've identified. And we can click on that and we can see that its name is meta-GRS, coronary artery disease. And it is original genome build, that is HG19 over here. And this is when it was released in the PGS catalog. It's by Mike Inoue. And then we can download the score here and get the variance and weights data file for this. And we can click this one here. The harmonize would just be trying to do the liftover from HG19 to HG38, but the current, it's okay if we are using like genotype data from HG19, we can use this one here. So we can download this guy over here. It clicks allow. And then we can see that it is a zipped file, and usually in Macs we can just quickly unzip it, and on Windows computers you can write gunzip, G-U-N-zip, G-U-N-Z-I-P, and then gunzip the file, or you can extract the contents from that scoring file. And you can see there are like a lot of SNPs here if we look at the scoring file, and it has like the header format that I was just showing you. It has the scoring file information, the version, the score, all of this information here. In the publication ID, and it has the SNPs, the chromosome number, position, effect allele, other allele here. So this is effect allele column, other allele is this other column that is not associated with the with coronary artery disease, and then effect weight, where a positive effect weight means that it is leading to increased risk of coronary artery disease, and a negative effect weight, for those SNPs that have negative effect weights like these ones, these are really good because they are associated with a lower risk of developing coronary artery disease. So there are a lot of SNPs here. It's huge, and I'm sure in other videos I could kind of go through this, but I want to focus, instead of having 1.7 million SNPs, I'll just focus on 21 just to make it a little bit simpler. But you can see there's a large scope of variance for computing, these polygenic scores. So this has a lot of variants, and we can keep scrolling down, but this is just a little snapshot of what this looks like. But yeah, you'll also sort of see how, for this example, they have effect allele and other alleles. And then in the simpler example that I will show, they don't have the other allele mentioned, but that just really depends. But they do have the effect allele and effect weight. But some score files will have effect allele and other allele information. Some may just have effect allele only and effect weight. I just wanted you to see that. So in this example, I'll be working instead on a simple score example, which is so much simpler. And it is the polygenic score given with the ID 53 here. So this is like the link, and we'll be going through downloading it and all of that. But essentially, it's for late onset Alzheimer's disease. And it is from a study from 2019 for 21 genetic variants, these SNPs, using the HG19 GRCH37 human reference genome. So Alzheimer's disease, it's a progressive neurodegenerative disorder, and it's the most common cause of dementia in older adults. So it's a brain disease that causes memory loss, cognitive decline, and behavioral changes. So Alzheimer's disease has two main forms. Early onset, which is like when you're younger than 65 years, it's usually rare. And it's caused by strong mutations in specific genes like APP, PSEN1, PSEN2. And what we're focusing on here is late onset Alzheimer's disease, or LOAD. It's the most common form of Alzheimer's disease, usually after age 65. And so Alzheimer's disease has these hallmarks, which is usually the buildup of amyloid beta plaques between the neurons in the brain, and these tau protein tangles that are inside the so these are plaques between neurons, and then there are these tau protein tangles that are inside neurons. So the plaques between neurons and these tangles inside neurons, and there's like brain shrinkage, especially like the hippocampus, which has our memories in the cortex. And so some of the symptoms are like memory loss, confusion, trouble with problem solving, mood and behavior changes, loss of independence, and there's no known cure. But some medications are out there. So it's a progressive neurodegenerative disorder leading to memory loss, cognitive decline, behavioral challenges. And it's sad because you start forgetting. People start forgetting and essentially regressing, becoming like babies again. So up next, we're going to show you how we downloaded our score file for late onset Alzheimer's disease from the PGS catalog, the polygenic score catalog. So let's kind of dive into it and see how did we download those scores, and just kind of show you a little bit more about it. So yeah, let's dive in, little guy. So now we're here on the pgscatalog.org, and we have these polygenic scores, and it was refreshed as of August, early August this month. So we're going to go here to polygenic scores, and we can browse through different traits here. And the one that I was looking at is related to late onset Alzheimer's disease. And here you can see that there's so many variants that are associated with these different traits and diseases. So what one we chose is the one that is the 53, which is on the next one here. So this is it here, Dostoevsky et al. Neurology, Alzheimer's disease, late onset. And so I used it because it has 21 variants. So it'll be like a little small case for us to use. So we can see that the original genome build is GRCH37. And then we can download the score here, which is where we go. And then we click this one here, and then we allowed it. And then basically what I do is I take the score file that we will get now. which looks just like this over here. I mean, the two is just because I've downloaded it quite a few times just to like make sure it all works before I made this demo. So that's the thing, and Macs automatically gunzip that compressed file because again we can have lots and lots of variants, so it compresses it. So what we do now is what I do is I just sort of take that and I migrate it, this text file. I just move it over to this score file folder that I have in Sanya demo. So we just have this score file here. And you can see again, these are the 21 variants that we have. These are RSIDs, reference SNP IDs, just to make it a little bit cleaner for you to understand what it looks like. Let me show you. If we throw it in and we thrash it into Excel, I don't recommend doing this when you have like a lot of variants. This is just because it's 21, so it's a little bit easier. So you can see reference SNP ID, chromosome number, position, effect, allele, effect, weight, and OR is for odds ratio. So this is the score file. Yay! And I've moved it into that folder for easy reference. Because I'm going to show you in step 2 how we organize that score file into separate files. So Alzheimer's disease, it's a form of dementia. You know, as I was mentioning, memory loss, cognitive disability, start forgetting, memory start fading, people start forgetting aspects of themselves. It's the 6th leading cause of death in the United States. Neuroinflammation, like the role of the immune system, it may play a key role. I've written a couple papers related to that. The heritability of Alzheimer's disease is rather high. It's around 80%, and there are various phenotypes out there that measure the extent and severity of Alzheimer's disease. So we can typically look at Alzheimer's disease progression from initial to moderate to severe stages. You know, there is resilience, psychological, that even if you have like Alzheimer's disease pathology and brain regions shrinking, you still kind of like have that cognitive resilience where you're not really showing signs of dementia. And that usually can show that if you keep up with your cognitive skills, you know, reading, learning, in socializing, playing board games, doing things that keep your mind healthy, eating well, maybe you could stave off some of the genetic effects of and risk factors for Alzheimer's disease that could happen. So that's what psychological resilience is, and people are studying that, like how can you kind of deter or delay the effects of Alzheimer's if it's meant to be. And then this epigenotype with the E4 alleles, they're the huge and the largest known risk factor for late onset Alzheimer's disease, though a lot is still unknown. And the CRED score for there's neuritic plaques, there's mini mental state exam scores to see how well you can do on like thinking and recall and memory. So in early Alzheimer's disease, what we typically see is mild memory impairment, especially like for recent events, difficulty with planning, problem solving, word finding, and then as it progresses, you know, confusion, disorientation, impaired judgment, behavioral and psychiatric symptoms, depression, agitation, hallucinations, and then late is severe memory loss, you know, dependence on care, you know, need full-time treatment, loss of motor function, incontinence, not able to go to the bathroom. So for instance, you can see this grandma here, like let's say that she's age 77, she's been healthy most of her life. At first she starts forgetting small things, where she put her keys or what she ate for breakfast, and over time she begins to repeat questions, struggles to remember names of close relatives, gets confused about the day or the place. Later on she finds it hard to manage daily tasks like cooking or paying bills, and she eventually needs help with dressing, eating, remembering family members. So Alzheimer's disease starts with mild memory problems and slowly worsens, affecting our thinking, our behavior, our daily life. So we want to know, like, you know, the genetic variants, like these SNPs, they contribute to Alzheimer's disease phenotypes, and what could be somebody's risk of having late onset Alzheimer's disease. So yeah, the polygenic score, the PGS, from the PGS catalog, is the single number that reflects someone's genetic likelihood of having a trait in a way. And the polygenic risk score is a single number that reflects someone's genetic risk for a disease. So they're just numbers that reflect somebody's genetic risk for a disease or a trait, and it's built by adding the effects of many SNPs, like tiny DNA changes, each with a small weight. So it's like a risk calculator. The more variance you carry, the higher your score. So also in the PGS catalog, you can see here that it might tell you the original genome build is this human reference genome, HG19, 21 variants here. And it was released in the PGS catalog in 2019. So yeah, as I show, like we click this, download score. Then we click this to download the text.gz file, and we click allow, and then it comes here as this zipped file, and then it gets extracted into this text file here. And then when we have the text file, it looks just like this here, for instance. So you could see that this is the genome build, GICH37 again. These are the headings for the PGS catalog. This is information about the name, the trait reported late onset, trait mapped, all this information, the citation, the paper. And you can see again Alzheimer's disease. So the most famous SNP region for late onset Alzheimer's disease is the APO gene. APOE4 allele is the strongest genetic risk factor for late onset Alzheimer's disease. Having one copy increases the risk, having two increases the risk even more. But APO isn't the whole story. Hundreds of other SNPs across the genome contribute small effects. And I was just showing a very simple example here, just 21 SNPs. But yeah, these SNPs are discovered in genome-wide association studies, GWAS, where scientists compare DNA from Alzheimer's disease patients and healthy controls. So late onset Alzheimer's disease, it's polygenic. It's caused by the combined effect of many SNPs, each with a small influence. And a polygenic risk score for Alzheimer's disease like this, for the late onset, it adds up these small genetic risks to estimate the overall susceptibility, given these SNPs and effect alleles. So at this chromosome, using this build, this HG19 build, so, based on the build, the positions, differ. It's kind of like translating different languages. So in a way, so we have like this position here, having an A kind of increases your risk of the late onset Alzheimer's disease. And this is the odds ratio. So whenever the effect allele is positive, the odds ratio is greater than 1. When the effect allele is negative, the odds ratio is less than 1. So as you see here, when it's negative, then the odds ratio is less than 1. And when it's positive, it's greater than 1. So these SNPs here that have a negative effect weight, they tend to be protective against late Alzheimer's disease, because their contribution, when we multiply them and we add them up to find the polygenic risk score, their contribution will be to lower the risk of late onset Alzheimer's disease. Essentially, this polygenic risk score is calculating the risk, somebody's genetic risk for late onset Alzheimer's disease. So if a SNP has a negative effect weight, it means that having that SNP will decrease your genetic risk for Alzheimer's disease. That's why it's protective. It's a good SNP to have. And if you have a positive effect weight, that means that having this SNP here would increase it for every copy you have from like one if it's from one parent, it increases it by 0.166. If it's from both parents, it increases it by 0.166 times 2. And if you don't have it, you don't have this being contributed to your score if you don't have this effect allele at this position in the chromosome. So that's sort of what it's trying to do. So people in, they calculate these polygenic risk scores, and they sum up these small genetic risks to estimate someone's overall susceptibility to late onset Alzheimer's disease. And people who have the highest polygenic risk scores may have several times higher risk compared to the lowest group. So that's what it's trying to do. And you can compare from the effect sizes from these genome-wide association analysis. So that study, those variants, they've come from here, from this paper in Neurology, as we see here, Tosto et al. So polygenic risk scores in familial Alzheimer's disease. And so this is the study that kind of released those 21 variants. So the objective was to investigate the association between a genetic risk score and familial late onset Alzheimer's disease and its predictive value in families multiply affected by the disease. So they talk about how they did this here. And again, this is the score file, but I've just kind of organized it. And these RSIDs, this is the SNP ID, reference SNP ID, chromosome position, effect, allele, the one that if you have it at this given chromosome at this position, if you have this, then it increases your risk of Alzheimer's disease. It contributes this much to it for each copy that you have from a parent. So yeah, this is sort of like how we kind of build out these polygenic risk score trait prediction models, from 23andMe, Ancestry DNA, different types of analysis, for individuals, like which mutations do they have, and then we kind of build out this polygenic risk score trait prediction model, set of SNPs and effect weights, and then we can use that to kind of predict in a model like somebody's risk of having that given condition. If it's a polygenic risk score, it's a disease. If it's a polygenic score, it's a trait. And so PGS catalog is focused on a variety of traits, including disease. But since I'm focused on Alzheimer's disease, I'm going to use PRS and PGS interchangeably.